So Madeline graduated from Concordia University in Edmonton in 2018 with a Bachelor of Science with high distinction. She then began her master's degree at the University of Alberta in Professor Stephanie Yano's lab, focusing on the immune response to pregnancy-associated malaria. Madeline has been awarded funding from several agencies, including CIHR and the Li Ka-shing Institute of Virology. Madeline is aiming to complete her degree in June 2021 and continue a career in health sciences. So welcome, Madeline, and share your screen.
Okay.
Great.
Everybody can see that?
Yep, looks great.
Okay, great. Well, good morning, everyone, and thank you for listening to my talk today. Today I'm going to discuss my research involving pregnancy-associated malaria and the immune response. So every year, approximately 125 million pregnancies are at risk for malaria. And when a woman becomes infected during pregnancy with the malaria species Plasmodium falciparum, this can lead to a number of poor outcomes, including maternal anemia, death, placental malaria, preterm birth, and low birth weight babies. So the malaria parasite invades red blood cells, and then they express attachment ligands on the surface of the infected red blood cell. This is so they may sequester throughout the body, mainly on the endothelium. In pregnancy, it expresses members of the PFEMP1 protein family. One of these we know is VAR2-CSA. This is a large 350 kilodalton protein that's made of multiple subdomains. And we know this ligand can bind to chondroitin sulfate A on the placenta or CSA. And by doing this, the infected red blood cells can. But if a woman survives multiple instances of pregnancy-associated malaria, she may acquire antibodies against these ligands, as seen at the bottom of the figure. These antibodies are capable of blocking the infected red blood cell from binding to the placenta, thus increasing clearance by the spleen and allowing monocytes to engulf these infected red blood cells. Usually, we see that multigravid women, or women who have survived multiple instances of PAM, do have better birth outcomes as opposed to primigravid women, or women in their first pregnancy. So our lab ran a previous longitudinal study in Colombia in South America, and we detected women who had submicroscopic infections of pregnancy-associated malaria. These are infections that cannot be detected using the typical microscopy methods, but instead need PCR. So usually in rural areas, these infections go untreated. So of the 187 women we followed, 45 of these women developed an SMI at some point in pregnancy. 27 of these were falciparum infections and 15 were vivax. Now, a number of these women also had mixed infections of both species, and some of these were simultaneous and some of these were sequential. So from this data, we collected serum at various time points during pregnancy, and we compared our antibody titer and functional data to actual pregnancy outcomes. And one of these were maternal hemoglobin. As we know, maternal anemia can be a common outcome of pregnancy-associated malaria. So a previous PhD student tested antibodies in our serum samples for this blocking function, so the ability to inhibit infected red blood cells from binding to CSA in vitro. And on the x-axis, you'll see percent inhibition. So this is how well the serum is blocking infected red blood cells from binding to CSA. And you'll see there is a positive correlation between this inhibitory activity and maternal hemoglobin, which was measured at delivery. So this led us to the question of what mechanisms may be in play where inhibitory VAR2-CSA antibodies can protect pregnant women from anemia. And this is where my research began. So we hypothesized in the placenta that when there are infected red blood cells in the placental, this leads to local inflammation. The malaria life cycle involves the infection and eventual lysis of the infected red blood cells, where merozoites then go and seek out other red blood cells to infect. And this cycle of obligate hemolysis does lead to inflammation that can propagate maternal anemia. And we think that, though this hasn't been shown yet, this could affect erythropoiesis systemically in the bone marrow, further propagating inflammation and anemia. But if a woman has antibodies, these antibodies could be functional, either with opsonic phagocytosis function and or blocking function. As well, antibodies are also capable of recruiting the complement system to further clear and lyse infected red blood cells. So we think that if a woman has acquired antibodies, this can aid from, first of all, blocking the infected red blood cells from binding to the placenta and encouraging engulfment of the red blood cells by phagocytes in the placenta. So my research work started by looking for complement activation in our serum samples. So the classical complement pathway is a long cascade of activating and lysing various components that eventually lead to the lysis of the infected red blood cell. But this begins with an antibody binding its antigen, and it's this immune complex that is required to recruit the first component of this pathway, which is C1q. And this is what I test for in vitro in ELISA assays, is the recruitment of C1q. So I began by looking at serum samples that were taken at enrollment of the study. So this is estimated to be around first to second trimester of pregnancy. And as you can see, there's a nice variation in C1q recruitment within our cohort. The numbers on the x-axis just designate our individual sample codes. And of our cohort of 42 women, we found that 30 were positive for C1q recruitment at enrollment. We then also looked at delivery, so serum samples taken from the mother at delivery, and we also tested these for C1q recruitment. And again, we found a nice variation of 24 samples, so approximately half the cohort was indeed seropositive for C1q recruitment. But of course, according to the classical complement cascade, there needs to be sufficient immunocomplexes forming to recruit the C1q. So then we then turned to the serology, and we looked for IgG and IgM seroreactivity within our cohort using an ELISA-based assay. So for set enrollment, we examined the cohort for IgG seropositivity, and we did see some nice variation again. We saw 21 samples that were both IgG and C1q positive. So we may be able to infer that IgG is capable of recruiting C1q. At delivery, we see this general decrease in IgG serology. So that leaves 7 individuals that are both IgG and C1q positive. And we've seen this in previous tests of the cohort, that by delivery, the anti-VAR2-CSA antibody titers have decreased. We also know that IgM is capable of recruiting complement, so we wanted to test our cohort for IgM seroreactivity. And we saw many samples that were seropositive for IgM, leaving 23 that were both IgM and C1q positive. So IgM may also be recruiting complement. With the IgM and the IgG data taken together, this left 2 samples that were only C1q positive. So we were seeing C1q recruitment, but we did not see the samples that were seropositive enough to pass our cutoff point. At delivery, again, we saw an overall drop, leaving 11 samples that were both IgM and C1q positive. And when we take into account our IgG data, again, this leaves 9 samples that are only C1q positive. So these 9 samples are neither IgG or IgM positive. So when looking at associations between the data sets, we see there is a slight positive correlation between the C1q reactivity and the IgG reactivity, but this correlation is lost by delivery. In contrast, IgM correlated strongly with, or more strongly, with C1q data at both time points. We see at both enrollment and delivery that IgM is correlated to the C1q reactivity, which we thought was interesting. But the main goal is to see if there's an association with our pregnancy outcome, which is maternal anemia. But after comparing all 3 data sets to maternal hemoglobin at delivery, we can see that there's no association between maternal hemoglobin and IgG, IgM, or C1q reactivity. So in conclusion, we find that C1q recruitment is not associated with maternal hemoglobin at delivery, but we do see an association with C1q and IgM seroactivity, which may be an interesting route to investigate. But perhaps more importantly is we're currently testing the functional activity of these antibodies. We believe a protective association may be seen in the antibody function rather than their titer. So as I showed in the beginning, we already have the inhibition data of the antibodies. And right now I'm testing for opsonic phagocytosis activity within this serum cohort. And then once we've gained this data, we aim to compare this to pregnancy outcomes and see if we see that positive association between maternal hemoglobin and opsonic phagocytosis, as we do with the antibody inhibitory activity. And with that, I'd like to thank my lab members, my committee, and my supervisor, and also the funding agencies. And I can take any questions.